A very good morning to respected teachers, seniors, and colleagues. Today I am going to present a journal article about nanotechnology in respiratory medicine. It was published on May 29, 2015. Over the past years, nanomaterials have found their way into more and more areas of life. Examples are new coatings and pigments, electronic devices, as well as cosmetic products like sunscreens and toothpastes. On the top of that, much effort is done to adapt nanotechnology for the treatment of human diseases. The term nano refers to structures in the range of 1 to 100 nanometers. In contrast to nanoparticles, which have to measure between 1 and 100 nanometer in all dimensions, nanomaterials may consist of elements bigger than 100 nanometer but need to be structured in the nanoscale and exhibit characteristic features associated with their nanostructures. In this context, the International Organization for Standardization defined the term nano-object as a material with 1, 2, or 3 external dimensions in the nanoscales. Nanomaterials have extremely high surface area to volume ratio. Therefore, some of them are very reactive or characteristically active. Moreover, in the nano world, quantum effects become visible and lead to some of the unique properties of nanoparticles, like viruses and cellular structures. Some nanoparticles are able to self-assemble to more complex structures. This makes them interesting candidates for novel drugs. On the other hand, it is necessary to redefine toxicology because of nanotechnology. Unlike classical toxicology, where dose and composition matter, in nanotoxicology, the focus has to be set on properties like morphology, size distribution, surface charge, and agglomeration state as well. Nanotechnology is important for respiratory medicine for several reasons. Firstly, it offers new approaches to treat diseases of respiratory tract. However, as nanotechnology uses in consumer products, cosmetics, and medicine is continuously increasing, it is also pivotal to understand potentially adverse effects of nanomaterials on the respiratory system. Additionally, studying respiratory effects of manufactured nanomaterials helps to understand the impact of combustion exhaust and ultrafine dust on human health. On the top of that, the lung is probably the most important gateway of nanoparticles to human organism. For the assessment of the safety in nanotechnology, it is therefore also important to elucidate which nanoparticle properties determine pulmonary resorption and biodistribution. Applications of nanotechnology in therapeutics and diagnostics. Although clinical application of nanotechnology in the therapeutics and diagnostic is still rare, there are multiple promising candidates for future use in the field of respiratory medicine. Drug delivery. Nanoparticles can act as vessels for drugs because they are small enough to reach almost any region of the human organism. Drugs can be bound chemically to the nanoparticles by a multitude of different linker molecules or by encapsulation. This allows better control of toxicokinetics. However, the main advantage is the capability of targeted drug delivery. The targeting can be active or passive. In case of tumor diseases, the leaky and immature vasculature of the fast-growing tumors can be taken advantage of in order to achieve passive targeting of chemotherapeutic loaded nanoparticles. This is called enhanced permeability and retention effect. The first generation nanodrug delivery systems rely entirely on the EPR effect. One example is Genoxol PM, a polymeric paclitaxel loaded polylactic acid block polyethylene glycol missile formulation. This nanocarrier has recently been tested in a phase 2 trial in patients with advanced non-small cell lung cancer. 43 patients were treated with the 4 3-week cycles of Genexol PM at 230 mg per meter square on day 1 combined with gemcitabine 1000 mg per meter square on day 1 and day 8. With a response rate of 46.5%, the therapy showed favorable antitumor activity. Moreover, immunogenicity was low. However, frequent grade 3-4 adverse effects like neutropenia and pneumonia were observed. The second generation nanoparticle drug delivery systems possess targeting ligands. This can be antibodies, aptamers, small molecules, and proteins. The attached ligands actively guide the nanoparticles and therefore the drug to tumor cells. The tumor-specific monoclonal antibodies are already widely used in cancer therapy. Those antibodies can be attached to nanoparticles for active targeting. In a recent study, polyglycolic acid nanoparticles that were conjugated with cetuximab antibodies for targeting and loaded with the drug paclitaxel were administered intravenously to mice with A549 Luke C8 lung tumors. The survival rate of these mice increased significantly compared to the control group. Another approach involves aptamers as targeting agents. Aptamers are synthetic oligonucleotides that are capable of binding specific target structures. Their small size, their simple synthesis, and their lack of immunogenicity make them promising ligands for nanoparticles. Moreover, small molecules such as folate can be used for targeting tumor cells that express a high density of folate receptors. In addition, tumors often overexpress receptors for several proteins. Proteins like transferrin, therefore, are common targeting ligands. These second-generation nanocarriers are already used clinically against lung cancers with substances like Orimmune CYT 6091 and BIND 014. Orimmune CYT 6091 is a drug delivery system based on gold nanoparticles functionalized with polyethylene glycol and tumor necrosis factor alpha. It has been used against adenocarcinoma of the lung in a phase 1 clinical trial. The TNF-alpha serves both as targeting and therapeutic agent. In this case, a phase 2 clinical trial for non-small cell lung cancer patients has been planned. The nanodrug delivery system by BIND 014 is currently tested in a phase 2 clinical trial as second-line therapy for patients with non-small cell lung cancer. BIND 014 nanoparticles consist of a polylactic acid core in which the anti-tumor drug docetaxel is physically entrapped. The particles are surface decorated with PEG to reduce elimination from the immune system and contain ligands against prostate-specific membrane antigen for targeting.
PSMA is expressed in prostate cancer cells and in the neovasculature of non-prostate solid tumors such as NSCLC. Preliminary data demonstrates that BIND 014 is clinically active and well tolerated. It also showed promising effects on patients with KRAS mutation, where ordinary antitumor agents usually fail. Additionally, adverse effects like anemia, neutropenia, and neuropathy were significantly reduced compared to solvent-based docetaxel. Nanoparticle-based drug delivery also offers potential in other fields of respiratory medicine. In experiments with tuberculosis-infected guinea pigs, it was demonstrated that inhaled alginate nanoparticles encapsulating isoniazid, rifampicin, and pyrazinamide showed better bioavailability and higher efficiency than other drug medication. Similar results were presented by Pandey et al. With three antitubercular drugs encapsulated in poly-D-lactic coglycolide nanoparticles. Moreover, another study demonstrated that perfenidone loaded nanoparticles have a higher antifibrotic efficacy in treatment of mice with bleomycin-induced pulmonary fibrosis than dissolved pirfenidone.
Hyperthermia. Nanoparticle-induced hyperthermia can be used to locally destroy tumor cells. Heat generation is usually achieved by two approaches, magnetic and photothermal hyperthermia. In magnetic hyperthermia, an extracorporeal coil creates alternating magnetic field that hits magnetic nanoparticles inside a tumor. This increases the temperature in the tumor without affecting healthy tissue. A recent study assessed the effect of inhalable superparamagnetic iron oxide nanoparticles in a mouse model of NSCLC. Compared to the non-targeted nanoparticles, the epidermal growth factor receptor targeted nanoparticles showed significantly more effective tumor shrinkage after magnetic hyperthermia treatment. The other approach, photothermal therapy, uses laser radiation in the visible or near-infrared spectrum and photosensitizing nanoparticles such as gold or graphene. A commercial product called Oroshell is available for tumor therapy. Oroshell nanoparticles consist of a silica core surrounded by a thin layer of gold. The gold nanoshells are administered intravenously and accumulate in the tumor due to the EPR effect. Upon exposure of the tumor to a near-infrared laser, the laser energy is efficiently converted to heat by the gold nanoshells. This therapy, which is called Orolase, is currently under clinical trials in patients with primary and/or metastatic lung tumors. Gene therapy. Like viruses, nanoparticles can be used as vectors for genes, but in contrast to viruses, they are less immunogenic and have higher DNA transport capacity. In a study, DNA-loaded polyethylene nanoparticles were used in order to treat lipopolysaccharide-induced acute lung injury in mice. After intravenous injection of the nanoparticles, the beta-2 adrenic receptor genes in the nanoparticles led to a short-lived transgene expression in alveolar epithelial cell. As a result, the 5-day survival rate improved from 28% to 64%. The severity of the symptoms measured by alveolar fluid clearance, lung water content, histopathology, bronchoalveolar lavage cellularity, protein concentration, and inflammatory cytokines was also significantly attenuated. DNA-loaded nanoparticles are also promising candidate in the treatment of cystic fibrosis. It was shown in a clinical trial that nasal application of DNA nanoparticles is safe and evidently leads to vector gene transfer. One major problem in this context is to overcome the mucous barrier. In a recent study, it was demonstrated that the densely PEG-coated DNA nanoparticles can rapidly penetrate extracorporeal human cystic fibrosis and extracorporeal mucous airway, mouse airway mucus. In addition, those particles exhibited better gene transfer after intranasal administration to mice than conventional carriers. Diagnostics. Nanoparticles have the potential to improve pulmonary X-ray diagnostics. Folic acid modified dendrimer and trapped gold nanoparticles were utilized as imaging probes for targeted CT imaging. In vitro and in vivo tests, the nanoparticles were trapped in the lysosomes of folic acid receptor expressing lung adenocarcinoma cells. It was possible to detect the tumor cells by micro-CT imaging after nanoparticle uptake. In addition, it was also shown that the particles possess good biocompatibility with no impact on cell morphology, viability, cell cycle, and apoptosis. Nanoparticles can also be used to enhance MR diagnostics of lung tissue. In experiments with intratracheal administration of gadolinium DOTA nanoparticles in mice, signal enhancements in several organs, including the lung, were measured with ultra-short echo time proton MRI. The signal change over time in the different organs demonstrated the passage of the nanoparticles from the lung to the blood, then to the kidneys, and finally to the bladder. Toxicological aspects of nanomaterials. Toxic effects of nanoparticles are a major concern in pulmonary medicine, especially ultrafine particles of low soluble, low toxic materials like titanium dioxide, carbon black, and polystyrene are overall more toxic and inflammatory than fine particles of the same material. This applies to both synthesized nanoparticles and natural dust. For nano-related toxicity, multiple mechanisms seem to be important. In the following, the interaction with the immune system, the creation of oxidative stress, and toxic effects on genome are taken a closer look at. In order to correlate toxic effects with nanoparticle properties, it is necessary to thoroughly characterize the selected nanoparticles prior to administration. Nanoparticle characterization. The most commonly used method to characterize nanoparticles for toxicology studies are transmission electron microscopy for size, morphology, and agglomeration, dynamic light scattering for the size distribution of the particles, zeta potential measurement for nanoparticle surface charge, and X-ray diffraction for particles crystal structure. In some cases, such as gold and silver nanoparticles, UV-Vis spectroscopy can be used to determine size and size distribution due to a spatial size-dependent optical activity. Ideally, nanoparticle characterization is repeated after administration, as changes of nanoparticles during the application process are possible. In vitro experiments, nanoparticles are usually applied by mixing with cell culture medium. The dissolved components of the medium, especially the ions, lead to agglomeration and precipitation of many nanoparticles, causing significant changes in their physiochemical properties. Similar effects are to be expected when nanoparticles come into contact with surfactant or other biological fluids. It was shown that some nanoparticles tend to form protein coronae in biological systems. Effects on immune system and inflammation. Many nanoparticles possess properties that give them potential to influence the immune system. In this context, nanoparticles' ability to penetrate cellular boundaries, to escape phagocytation by macrophages, to act as heptans, and even to disrupt the Th1 Th2 balance might be essential. For carbon black nanoparticles, a recent study investigated the effects of inhalative exposure on mice with bleomycin-induced pulmonary fibrosis. The analysis of histology as well as cytokine expression suggested that the nanoparticles triggered an inhalation exacerbated lung inflammation. The author concluded that, especially for people with pulmonary preconditions, inhalation of nanoparticles can lead to serious health problems. In this context, another study found out that pegylated cationic shell cross-linked candle-like CSCK nanoparticles produce significantly less airway inflammation than non-pegylated ones. This was explained by a change in endocytosis. In contrast to the clathrin-dependent endocytosis of non-pegylated particles, the pegylated CSK nanoparticles showed a clathrin-independent route. On the other hand, some nanomaterials exhibit impressive immune modulating activity. As an example, gadolinium compound, a fluorine derivative with a gadolinium atom inside, showed anti-cancer activity without being cytotoxic. In vitro studies demonstrated that gadolinium compound activated dendritic cells and even induced phenotypic maturation of those cells. Moreover, the gadolinium compound treated disease also stimulated erogenic T cells in a Th1 characteristic. The effect of gadolinium compound was comparable, probably even stronger than the effect of lipopolysaccharides on disease. The study also verified that the nanoparticles were free of LPS contamination. In vivo experiments on ovalbumin immunized mice showed enhanced immune responses comparable to the adjuvant effect of alum on ova mice. However, whereas alum led to a Th2 response pattern with IL-4, IL-5 and IL-10 upregulation, gadolinium compound caused a Th1 pattern with upregulation of INF gamma. Similar results were demonstrated in other study using a murine asthma model. OVA sensitized mice that were additionally treated with a nanomaterial graphene oxide during allergen sensitization had stronger airway remodeling and hyperresponsiveness responsiveness than mice that have only been treated with OVA. The graphene oxide lead to a downregulation of Th2 dependent markers such as IL4, IL5, IL13, IgE, and IgG1, but increased Th1 associated IgG2A. Moreover, the graphene oxide increased the macrophage production of mammalian chitinase 3-like protein 1, and AMCase, which could be the reason for the overall augmentation in airway remodeling and hyperresponsiveness. However, this kind of immune modulation can also be utilized for therapeutic purposes. In a recent study, a nanoparticle-based vaccine has been used to treat dust mite allergies in mice. The immune-modulating carriers were generated by loading dust mite allergen DERP2 and the potent Th1 adjuvant unmethylated cytosine phosphate guanine into biodegradable polylactic coglycolic acid polymer particles. Mice treated with those nanoparticles showed significantly lower airway hyperresponsiveness as well as lower IgE antibody levels after a 10-day intranasal DERP2 installation compared to the control group. The authors conclude that this biodegradable nanoparticle-based vaccination strategy has significant potential for treating HDM allergies. Oxidative stress and catalysis. Oxidative stress is often brought in context with nanotoxicology. It can be measured directly with dichlorofluorescin or indirectly by the upregulation of reactive oxygen species eliminating enzymes like superoxide dismutase. Another approach involves test whether the nanoparticle dependent toxicity can be reduced by the application of an antioxidant. Widely used semiconductor materials such as lead sulfide nanoparticles may have the potential to generate oxidative stress in the lung. A recent study tested the toxicity of intratracheally applied 30 nanometer and 60 nanometer lead sulfide nanoparticles on rats. Oxidative damage was
Evaluated based on superoxide dismutase, total antioxidant capacity and concentration of malondialdehyde. In addition to inflammatory responses, both 30 nanometer and 60 nanometer groups showed increased oxidative damage compared to control groups. The effect was significantly stronger for the 30 nanometer lead sulfide compared to the 60 nanometer nanoparticles. And another nanomaterial which is associated with oxidative stress is nanosized titanium dioxide. Lee et al. Induced pulmonary injury in mice by daily intranasal installation of suspended 294 nanometer titanium dioxide nanoparticles for 90 days, demonstrating that the rate of reactive oxygen species generation increased with increasing TiO2 doses. Moreover, lipid, protein, and DNA peroxidation products were identified in elevated doses, which suggests that ROS-dependent lung damage was significant in the nanoparticle-treated animals. Furthermore, in vitro tests on BEAS2B and A549 lung cell lines demonstrated that the commonly used nanoparticles, zinc oxide and ferric oxide, are very different in terms of creating oxidative stress. The ferric oxide nanoparticles with an average diameter of 39 nanometer were distributed in the cytoplasm, whereas 63 nanometer zinc oxide nanoparticles were trapped in the organelles such as endosome. In contrast to the ferric oxide nanoparticles, the zinc oxide nanoparticles caused reactive oxygen species production as well as cell cycle arrest, cell apoptosis, mitochondrial dysfunction, and glucose metabolism perturbation. Genotoxicity. Another important type of toxicity caused by nanoparticles is genotoxicity. A common method to quantify genotoxicity is the COMET assay. which uses electrophoresis to detect DNA strand breaks. This assay was used in a recent study to check whether intratracheal installed fluorine C60 nanoparticles induced DNA damage in the male rats. However, despite inflammatory responses and hemorrhages in the alveoli of the C60 treated rats, there was no significant increase in fractured DNA in their lung cells. Therefore, it was concluded that even at inflammation-inducing doses, fluorine C60 nanoparticles have no potential for DNA damage in the lung cells of rats. Similarly, another study demonstrated that intratracheal installation of anhydrous titanium dioxide nanoparticles on rats did not result in genotoxicity. None of the titanium dioxide groups showed an increase in fractured DNA, while the positive control with ethyl methanosulfonate exhibit significant increases. In contrast to those results, Joska Zhou et al. Found that even in low doses where no inflammation occurs, Printex 90 carbon black nanoparticles induced genotoxicity in mice. There was no inflammation, cell damage, and acute phase response, which means that the increase in DNA strand breaks are related to direct DNA damage caused by the nanoparticles. On the other hand, a recent study suggests that CEO2 nanoparticles may be even used as antioxidant and anti-genotoxic agents in the lung after treatment with the oxidative stress inducing agent KBRO3. BAS2B cells pre-treated with CEO2 nanoparticles showed significantly less intracellular ROS as well as reduction in DNA damage compared to non-pre-treated cells. Biodistribution, nanoparticle detection. Research on biodistribution of nanoparticles requires tracking of the applied nanoparticles in the test animals. Conventional light microscopy is not able to detect nanoparticles because of Ebb's law. Therefore, electron microscopic imaging is often required. However, light microscopy can be used to describe the nanoparticle-induced changes in the cell morphology without being able to see the nanoparticles themselves. Additionally, nanoparticles can be indirectly made detectable in the light microscopy by a method called autometallography. This is a silver staining that can be used to increase the size of several types of nanoparticles like gold, silver, and some metal sulfides and selenides in the histological section. This technique was used to detect silver nanoparticles in the olfactory bulb and lateral brain ventricles of mice that had been intranasally treated with 25 nanometer silver nanoparticles. Particle deposition and resorption in the respiratory tract. Most research about biodistribution of nanoparticles in organisms focuses on intravenous injection. However, nanoparticles were shown to be able to pass the blood-air barrier of the lung. Whether or not nanoparticles can travel through the lung into the body seems to be size-dependent. This was evaluated by injecting neutron-activated radioactive gold nanoparticles of 1.4 nanometer to 18 nanometer intratracheally to rats. The bigger nanoparticles almost completely retained in the lung, while the significant amounts of the smaller 1.4 nanometer particles were found in blood, liver, skin, and carcass for 24 hours after installation. Choi H.S. et al. Applied nanoparticles of different size and charge to mice. The nanoparticles were tracked in different organs through fluorescence labeling. It was demonstrated that nanoparticles rapidly translocated to the mediastinal lymph nodes if they possess a hydrodynamic diameter of 34 nanometer or less and a neutral or anionic surface. Bigger and positively charged nanoparticles exhibited no significant uptake. In addition to physical parameters of the applied nanoparticles, the health status of the exposed organism also seems to play an important role. A recent study showed that the distribution of oropharyngeal instilled 40 nanometer gold nanoparticles is influenced by additional LPS treatment. The gold content of organs was measured with inductively coupled plasma mass spectroscopy (BL BALB or BALC) mice that had been oropharyngally treated with LPS 24 hours prior to the nanoparticle administration exhibited less gold content in their lungs than untreated mice. In both groups, gold was detected in different organs. High concentrations were found in heart and thymus in the non-LPS group, while the LPS-treated mice accumulated most of the gold in the spleen. The author concluded that nanoparticle uptake may depend on medical preconditions. Conclusions: over the last decade, major breakthroughs in nanotechnology have been achieved. It is only a matter of time before new nano-based drugs reach respiratory medicine, especially the fields of targeted drug delivery, gene therapy, and hyperthermia offer great potential for modern drugs. On the other hand, the increased use of nanomaterials in all fields of life also bears risk of exposure through inhalation. It is therefore essential to understand pulmonary toxicology of nanomaterials in all its facets. However, it is still very unclear why the toxic effects of nanoparticles in the respiratory tract is so inhomogeneous and not well predictable. In this context, not only local reactions of lung and airways, but also nanoparticle uptake and distribution in the organism are important factors and therefore fields of current research. As only few nanoparticle compositions have been tested, it is questionable whether those results can be easily adapted to other nanoparticles. Because of the continuously increasing diversity of engineered nanoparticles, toxicology can hardly keep pace with the safety assessment and of future products. Therefore, more attention should be set on this wide field of research. These are the references for this article. Thank you.